Greetings, parasite ecologists. Now that you've gotten to know the cestodes a little bit, let's talk about the habitat they use as adults, which is the vertebrate gut. We've discussed this before. Remember, we talked about the energy efficiency of parasites and the rate at which they convert the food they consume into parasite biomass. Back then, we talked about Hymenolepis diminuta, the rat tapeworm. You'll remember that we discussed how the rat is super inefficient at absorbing energy, with 95% of the total energy it absorbs going to maintenance and only 5% going to the production of new rat biomass, either body growth or reproduction. Meanwhile, the rat tapeworm, infecting that rat, allocates 63% of its energy to metabolic maintenance, while 37% goes to production, showing a much more efficient conversion of absorbed food into biomass. Now, how does this tapeworm manage to do that? We're going to answer that question right now. I mentioned that H. diminuta is a well-used model system and that there's been a lot of research on rat tapeworm growth and physiology. The H. diminuta life cycle is a pretty simple one, at least as far as tapeworms go. H. diminuta begins life in the intestine of a rat, where it does sexual reproduction and produces oncospheres, or eggs. The oncospheres are dispersed in rat feces, which are then consumed by an intermediate host, like a flower beetle. There can be a few different species of beetles involved, but in this case, we're looking at the flower beetle Tenebrio molitor. If this beetle looks familiar to you, it's probably because you've worked with mealworms before. Mealworms are a common thing to see in science classrooms. They are Tenebrio molitor, the larval stage of the flower beetle. If you were to leave your mealworms at room temperature for a couple weeks, you would wind up with a container full of these flower beetles. Once that flower beetle consumes those Hymenolepis diminuta oncospheres in rat poop, it becomes infected with the larval stage of the parasite, called a metacestode. When that beetle is eaten by a rat, the life cycle is closed and completed. The metacestode grows into its adult form, mates, produces eggs inside the rat's intestine, and it starts the life cycle all over again. One of the ways that tapeworms are so efficient at using the energy that comes to them is through structures called microtrichas. The singular form of microtrichas is microthrix. These are invaginations of the tapeworm's tegument or body wall, analogous to microvilli in the human intestinal lining. You know how the surface of the intestinal epithelium is complicated, with lots of invaginations to increase the surface area available for absorption. Tapeworms have something similar, but on a much smaller scale, and they're called microtrichas instead of microvilli. They increase the absorptive area of the tapeworm's body surface. Let's compare the amount of absorptive surface between the rat and its tapeworm. If you cut a rat's intestines out, lay them out totally flat, and calculate that intestine's total absorptive surface area, the rat has about 90 square centimeters of effective absorptive area in its intestine, which translates to about 9 square centimeters per 100 milligrams of rat intestinal biomass. The rat tapeworm's effective absorptive area is 390 square centimeters, or 39 square centimeters per 100 milligrams of tapeworm biomass. So, the rat tapeworm has a greater surface area overall and a greater surface area of intestine per milligram of body mass that intestine has to feed. How is the rat tapeworm able to have so much more surface area? It's because the rat gut is multifunctional. The rat gut needs to do things like secrete, digest, absorb, ensure things move through it, and perform some immunological defense. All the tapeworm has to do is absorb. Tapeworms literally lay in a bath of perfect food all day long. They barely even have to move. The tapeworm has almost no functions to perform except absorption, so it can invest really heavily in having a body surface finely tuned for absorption, whereas the rat intestinal epithelium needs to perform multiple functions. This is part of the reason why tapeworms are so much better than rats at absorbing and converting food into biomass. What is the tapeworm actually consuming while it's inside its definitive host? Carbohydrate is the most limiting resource, and that includes glucose and galactose. Fatty acids, which can be reduced in the host by up to 30%, are also consumed. Proteins are generally not limiting for the tapeworm because the sloughed off host cells, those dead cells that fall off the host's intestinal epithelium, they provide a sufficient source of the amino acids that are important building blocks of proteins. And as for water, the worm can interfere with the host's water absorption, causing diarrhea. But it's not all sunshine and roses for the tapeworm because the vertebrate gut is not consistent across its length in terms of its quality as a habitat. Consider this diagram of the vertebrate alimentary tract, moving from the mouth down to the esophagus, to the stomach, small intestine, large intestine, and anus. Across that gradient, many things change, including the pH. pH is about 2 in the stomach, 7.5 in the duodenum, 7.7 in the small intestine, and 8 as you get closer to the colon. This change means that the position of the worm within the vertebrate gut is crucial. It determines both the environmental conditions like pH and the resource availability. What's going to be there for the tapeworm to consume as it lays bathed in food? Take sharks as an example. This is a dissected shark with the anterior end toward the right side of the slide and the posterior end toward the left. You can see the stomach in the lower portion of the slide, which doubles back up and then goes into the top of the spiral valve intestine, emptying out at the rectum and the anus. The spiral valve is a special organ unique to sharks. It is a super compact way to have an intestine, ensuring that the shark remains hydrodynamic while still having a lot of surface area for absorption. It's kind of like a spiral staircase, but in intestine form. The spiral intestine in sharks has very different conditions in the anterior portions compared to the posterior portions. On this plot, you'll find the distance along the alimentary canal from the stomach to the anus, represented on the x-axis. On the y-axis, you'll see the pH. You can see that the anterior-most portions of the spiral valve have pretty low pH, which might be an environment that a parasite that doesn't want to be dissolved should avoid. At the same time, the resources available also change across the spiral valve. Here's a plot with distance along the alimentary canal from the stomach to the anus, represented on the x-axis. On the y-axis, you're going to see the carbohydrate concentration. The stomach has very few available carbohydrates, but as soon as you enter the spiral intestine, carbs increase as you move closer to the anus of the shark. As a result, tapeworms that live in these shark guts engage in some habitat partitioning. We find one type of tapeworm with the highest abundance in the stomach, and then 3 more that have their favorite areas to inhabit within the shark spiral valve. This is probably due to a combination of factors. The tapeworm living with its scolex stuck in the stomach probably takes advantage of its specialization in dealing with the extremely low pH of the stomach, a pH that no other tapeworm can tolerate. The other 3 tapeworms are probably making compromises between the pH that they're physiologically capable of tolerating and the types of resources that they need. They're also avoiding one another. There's probably some competition for territory among these different tapeworm species. How does competition within the vertebrate gut happen? Let's find out. I'll see you in the next video.